So the next presentation is by Altan Kokatulum. So Altan, wherever you are ready.
All right, can you all see and hear everything okay?
Yes.
Okay. All right, hello, my name is Altan Kojatalam. I'm from Alfred University, and this is my REU project on improving ALPACA, which is a program standing for Automated Landmarking through Point Cloud Alignment and Correspondence Analysis, which will make a bit more sense soon. My mentor for this project was Dr. Porto from the biology department at LSU. So this program has to deal with the field of geometric morphometrics, which is analytic techniques that biologists use concerning specific anatomical points of interest on a structure. As you can see here, we have two different skulls, which have all these little points on them. So they will take the coordinates of those points and do various mathematical analyses with them to test hypotheses about how these structures evolved and how they function within the ecosystem. So how the program essentially works, it is to automatically put those points on the structures. Usually people would do that manually, which can take a while. And so this is a way to make the data acquisition part of these studies much easier and faster. So how ALPACA works is we start with two samples, one of which has been annotated manually, has all the landmarks on it, and then we have a second one which we want to get the landmarks for. So we have our two samples here, and then we are going to downsample them to get point clouds instead of the full 3D mesh. So then we're working with much less data, which allows the program to run faster, and it doesn't seem to have a significant adverse effect on the accuracy. Then it does a rigid alignment, which is essentially rotating and translating the structures until they are lined up as best as possible. And then the deformable or non-rigid alignment here, it will sort of squeeze and stretch the initial source model, which has the landmarks, to match the model that we're trying to get landmarks for, so that the shape is matching the second one, but all the landmarks are preserved. And then it will just transfer the landmark points onto the new model. And so that non-rigid point cloud registration step takes the longest. It is a very significant mathematical undertaking. So that is the main point we are trying to improve here. It uses a coherent point drift algorithm, which imposes constraints on the points in the cloud so that they can only move in specific ways, and that one point can't move too differently from the points around it. And it's based on some obscure mathematical concept called Gaussian mixture models. We don't have time to get too into that. So the change that we made, there was a new coherent point drift algorithm that was published just last year, and it runs in a Bayesian setting. Essentially means that the underlying assumptions are looser, which allows us to take some shortcuts in a very broad sense. So we can accelerate this with various methods in linear algebra with the Nystrom method and KD tree search. And here we can see that we get to similar levels of error in about the same steps, but it is much faster. The BCPD is the new one, so the orange lines have a much faster computing time than the old method, which are the blue here, and also scale better with the size of the point clouds. Like this one is with 100,000 points, whereas this one is the new method with 100,000 points. So there's a much bigger increase from 50,000 to 100,000 here than between these two. These are what the algorithms look like, but we definitely don't have time to explain those. So this is what the program looks like. We have our 3D model here. You can see these little pink dots on there are the landmarks. You can rotate it around, look at it however you want. Over here is where you tell it what you want it to do. So we can pick out the source file here, the landmarks for the source file, and then a folder for all the targets that we want to find landmarks for. and then the folder we want to put our outputs in. There's a few advanced settings you can fiddle with, but it's pretty straightforward. You just pick out your files and then you click the run button, and you can see all the magic happen over here. And this is within 3D Slicer, which is an open source platform used in all sorts of biological modeling settings. And it's free, relatively lightweight. It's handy for a lot of things. And then since we were able to get it, use this faster algorithm, we are then able to run the comparison between multiple source models for each target. So instead of just running one pair, we run the same target with 5 different sources. And that allows us to get more accurate results because of the variation between the source models and the targets and all that. And so essentially we just run the whole pipeline for the 5 different sources, and then we take the median of the results. And we can see here that that improves the accuracy. This is a graph of the error versus the number of sources. So here at 5 sources, we can see that the error is quite a bit lower than for 1 and 3. The variance is also somewhat lower, and that going to more than 5 sources doesn't seem to help much. So 5 is the sweet spot there. Here's a more detailed comparison of accuracy. We see the median error over here for the original version is a little bit higher than the median error over here for our new version with the new algorithm and using 5 sources. So this is for each landmark in our data set. Each sample has 45 different landmarks in it. So each box here is the error for each landmark. Then the different colored points here are to compare with other methods. So blue is another automated method that has been published. We're generally under there, which is good for us. And then green is the error that you would expect for different people putting the landmarks on manually. So we're taking that as the minimum we could possibly hope to achieve. And we're doing pretty well in that capacity. Also worth noting that, again, the variance is lower as well. It's not just the median error with the new version. Here is more showing how we can go faster with larger point clouds using the new algorithm. As you see, the slope of those is much less than for the original here. So sometimes it's possible that using a larger point cloud would be helpful. In the data set we've been using, 5,000 is sufficient, but if you have a more complicated model, we might need more, and it's nice to have that option. And yeah, that's what I've got. Any questions?
So this will be working for any kind of bone and any species?
Yes, you can take any structure as long as you have one that you have manually annotated landmarks for, and then another file that you want to find the landmarks for, as long as they represent the same structure. You can do that here. There are certain limits in how much variation we can have that the program will still have results that make sense for, but largely, yes, it should work for any structure.
We can pass to the next person, Janice Campbell.
Okay, so hello everyone. My name is Janice Campbell, and I'm a student here at LSU. Over the past few months, me and Dr. Nam have been working on a project called Farmware. And with that, I'll get right into the introduction. So I've worked on mainly 2 projects during my time at this program, one of which being Farmware, which is what I'll be focusing on today, and the other being the wearable GPS. I just want to briefly mention it because I did work on it, I did put time into it, but it's still a work in progress, but it does fit into the overall theme of the work. The basis of this research comes from previous studies about the mental health benefits of nature during stressful times. The concept of biophilia is rooted in historical and anthropological evidence of the human-plant relationship. Because this relationship is so well documented, that made it much easier to understand the immediate benefits of the symbiosis. But what is not as well documented is the possibilities that technology presents to this. So the next evolutionary step of this idea uses embedded and embodied interactions to enhance the direct experience with nature. And by having the user actively participate in the development of a plant, as well as having a physical attachment to the plant, the goal is to provide a more tactile means of caring for nature. And hopefully by caring for plants, the person is more conscious of themselves as well. So going a little bit deeper into farmware, the initial idea was to develop a closer relationship with our food, but as we'll see later, this quickly evolved in a number of different directions. Despite this, there are a few fundamental truths that persisted through development. 1, emphasis on DIY, 2, practicality, and 3, sponsoring an emotional relationship between nature and humans. So because DIY is such a big component, it was important that the designs were highly modular and easily customizable. A little bit about the back end of stuff. I modeled everything in either Maya or Rhino and used both STL format for printing and OBJ format for usage on the website, which I'll explain shortly. Everything is 3D printed using TPU, which is a flexible rubber-like plastic. Of course, the plants themselves presented a lot of design challenges. We tried a lot of different plants, from arugula to basil, even catnip. Most of these were microgreens, meaning they were edible, which fits into our earlier hypothesis of developing a personal relationship with your food. Because of this, an iterative approach was vital for development. We tracked the growth of each type of seed in a journal over roughly 20 days, just to understand how each plant behaves and grows. Naturally, things get a bit more complicated in practice. One of the earliest designs was the Bengal that you see on the top right. But the problem with that is that water would not stick to the surface. It would just roll off and fail to hydrate. And of course, when seeds can't hydrate, they can't form roots, and thus they never grew. I also suspect that these are a bit too shallow, so roots couldn't form there either. So because interaction is critical to this project, as well as having everything easily accessible for a variety of users, with this in mind, a web app seemed perfect for these goals. Everything is built in JavaScript with the help of a few libraries for some of the more complicated stuff like UI, machine learning, 3D rendering, the STL exporting, and procedural graphics and sound. So there are 3 parts to the website. The first being the design section, which allows users to design their own firmware and export to an STL format for printing. The modular design that I mentioned in the individual components makes it easy to not only create custom firmware, but also to load in these components to be switched out as needed. The designer loads in the base model, and the user can control certain parameters depending on the type chosen. Next is the monitor tab, which uses machine learning to classify the health of the plant. It reports on 4 total states: seeds, healthy, unhealthy, and empty for the base container. And finally, the play tab takes the green values per pixel, using some filtering, of course, because you don't want every pixel returning a value, and draws a green dot and plays sound depending on the amount of green. This feature simply gives the user another option to interact with their plant in a manner that's not so life or death. And this hopefully encourages further motivation for taking care of the plant. So once development was in a presentable state, we moved on into user trials. The purpose of the user tests was to simultaneously understand the practical usage of firmware, as well as get general feedback for the system itself. The trial consisted of a few stages. The initial survey was to get an understanding of the user's relationship with nature and agriculture, with questions like, do you feel close to nature? Are you concerned about food waste? How much time do you spend outdoors? After the survey, the user would then design their own custom wearable, which we would print back at the lab and return to them. After they chose and planted their seeds, they would officially begin the trial, spending upwards of 2 weeks with their new device. In the final interview, users would revisit these questions and concepts and offer any insight into the process of taking care of their firmware. They would also retake the initial survey so that we can compare answers later for our data. While the user trial was running, we also had a series of quick interviews with professionals from various fields, such as apparel design and merchandising, wearable tech, and digital media. These discussions gave us deeper insight into the functionality and design of the project as a whole. These interviews will be analyzed using a method called grounded theory, which is a part of a methodology that inspects patterns from data. These interviews would later be analyzed for specific keywords. And the results of this would also be compared later to the user test results. So the users came from a variety of backgrounds, so we definitely got some interesting feedback. On average, users would use firmware for about 2.4 hours for roughly over a day over the course of the 2 weeks that they had it. One thing that really surprised me, even when I was trying it myself, was how easy it was to get emotionally attached to the plant. The act of caring and sponsoring development certainly had an impact on how successful this project was. And many users also reported that they felt similarly. Possibly the most powerful effect of this project was the power of seeing things in everyday life that could contribute to bettering our relationship with the world. Beyond the idea of just agriculture, users also found power in the social, educational, and sensory elements of farmware. Our results tell us that users saw many more applications than was originally envisioned, which is great for future development. So, as stated earlier, the concept had evolved to be much more encompassing, and this was reflected in the discussions from our users and experts. One thing that consistently came up was requests for an app that would track the plant's water, sunlight, and nutrition, which would then translate into the user's needs for water, outdoor time, and healthy food. That certainly wasn't an early idea we had, but due to time constraints and like the amount of data that would need, this was scrapped in lieu of the website. But that definitely be a more long-term goal for future work. Naturally, we want to take the design feedback and further develop firmware with new plants, technology, and materials, like possibly using edible mushrooms or mycelium as a base. We can also take this from the micro scale to the macro scale and use this in larger scale generative work. But going forward, we will take these findings and start developing our paper within the next month, with submission to a major HCI conference following soon after. So these are a few of my references, and with that, are there any questions?
Yes, I wonder, I mean, you say to do it with mushrooms, but mushrooms are much bigger. So what kind of wearable can you create?
Yeah, that's, I mean, that's very interesting. Like there's certainly like, we can do hats, we can do like, full on like vests or something. Like it's not limited to just like jewelry. But I'm sure there's like, there's like smaller scale mushrooms. I'm not a mushroom expert, but even if we don't use it like as the plant itself, we can use kind of that structure as the base for, instead of like the TPU plastic that we were using, we can definitely use like the mycelium itself for the base. So that's just another option.
Is there anything really surprising, like out of all results? Is there anything really surprising for you?
I think the most surprising thing for me, I was really surprised that people were so attached to like the smell option. Because that didn't, like when I was smelling the basil and the arugula, like oh, it's really strong. Like there's like potential there. But also I was like personally surprised by the emotional attachment component, because I've never grown plants before. I've never like cared for plants all that much, but seeing how that resonated with a lot of people, like that was really, that was further motivation for me. Yeah.
Any other question or suggestion?
Do you foresee this wearable things to be on the market soon, or?
Yeah, that was we definitely talked to a lot of people about that. If this were a product on the market, it definitely be in like some kind of kit. But yeah, I think with like further refinement, this could be a potential product if we decide to go that direction. Yeah.
But all of us, we will get one piece to try, no?
Of course, of course.
So let's move. Next is Aaron Valdez.
Okay, so let me start. Okay, so today I'm going to be talking to you about optimized multi-layer structures with phase change materials. My name is Aaron, and my mentor is Dr. Verones. So, have you guys ever wondered how privacy glass works? Well, it all boils down to the idea of switching the transparency of the material from transparent to opaque when you apply an electrical voltage across them. And the most common material for privacy glass is PDLC, which stands for polymer dispersed liquid crystals. So how does all this stuff that I just mentioned related to optimizing multi-layer structures? Well, the simple answer is that on our research, we try to come up with combinations of multiple phase change materials as a way to obtain the biggest difference in absorption. Why are we looking for the difference? Well, as seen in this clip from the privacy glass, the material is able to change its absorption from probably 20% to 100%, which means there's an 80% difference, probably turning from 20% to 100%. So as you can see from this diagram over here at the bottom, the phase change materials that we are mostly testing is GST, which stands for germanium antimony tellurium. The main groundwork that has been done in this project was done by graduate students of Dr. Verones, which created a program which is able to not only find the most optimal thickness, but also the optimal combination of material and thicknesses to obtain near perfect absorption. This was done via hybrid optimization algorithm coupled with the transfer matrix method to optimize material composition and the layer thicknesses of the aperiodic multilayer structures. This innovation led to the previous REU student to expand its application. He worked on optimizing their absorption ratio in multi-layer structures when GST switches between its crystalline and amorphous phases. This is where I came in. My first step in this project was simply being able to run the original code in the HPC computer and see if my results matched the work of previous students. Next, I created a method to obtain two optimized results for both scenarios, a crystalline state or amorphous state. Then I have to implement a fitness function which will obtain the maximum of the difference between the absorption of the crystalline state and the amorphous state. Finally, my last step was to run as many simulations as possible to see a pattern and test the accuracy of our data in place of the physical experiments that we would do. Here's the latest simulation of a 5 multi-layer structure run by me, and here's a simulation of the original paper with 5 multi-layer structures. As you can see, the spectrums are the same. Here we can observe the material composition and the layer thicknesses are the same, which means that my results are consistent. So to finish the previous work, I created a function that converts all multi-layer structures with GST of any type into either only amorphous or only crystalline. Then I devised a fitness function to observe how well each multi-layer structure performs based on their difference and they're in the ratio of absorption. So the strategy to convert multi-layer structure with GST of any type into either only amorphous or only crystalline ones is by swapping all GST crystalline layers with GST amorphous layers or vice versa. As seen in this code, we first find the material ID of GST amorphous and then we iterate through all the layers, switching those layers that have crystalline with amorphous. Then as you can see, we find all the GST crystallines and finally we just swap them. And it works in the same manner when switching from amorphous to crystalline. The difference in this function. As in this code, we first convert original multi-layer structures into two new multi-layer structures consisting of either only amorphous or only crystalline. Then we simulate each of those new layers that we created. As you saw from the previous slide, this is a result of the simple conversion of our current multi-layer structure to two different multi-layer structures made of either only amorphous or only crystalline. Next, we have to simulate each of those new multi-layer structures created. As you can see by the result 0, it will be the result of the simulation, and result 1 will be the result of the simulation of crystalline. This simulation uses a fitness function that iterates through all the specified wavelengths to find the maximum absolute difference of absorption between the two phases. And as you can see, we're actually finding the maximum absolute difference of the absorption of the crystalline state and minus the absorption of the amorphous state for all the wavelengths. And by the way, 500 here is an arbitrary value for maximizing our results. So the ratio fitness function. This fitness function is similar to the previous one, except that we're looking for the ratio of absorption. More specifically, we're looking for the maximum ratio of absorption of crystalline over the absorption of amorphous across all wavelengths. And here I'm going to describe the fitness function, which is basically we iterate through all the wavelengths, then use a comparison to get the maximum, and finally here we are using arbitrary values to be able to maximize our results. So yeah, and as you can see, our main goal is the ratio fitness function is to obtain the maximum ratio between the absorption of crystalline or the crystalline state over the absorption of the amorphous state for all the wavelengths. So for the simulations, there were two phases for the simulation. First, testing the code. This was to see if the result is consistent. The second phase was obtaining results of fitness functions for the difference and ratio between amorphous and crystalline. Are the results consistent? Well, yes. For the difference fitness function, you can see that all the spectra are identical, which means that all results are consistent. The same happens with the ratio fitness function, where we're also able to obtain the same spectra for the same amount of layers, which means all results are consistent. Difference fitness function, relevant results or optimal results. In this chart, we can see how increasing the number of layers increases the difference in absorption between amorphous and crystalline. As you can see, there's a visible difference between having 2 layers and 7 layers. In this graph, I present the most optimal result with 0.65 difference in absorption. This was obtained with 15 million trials for 2 layers. By the way, these are preliminary results because most likely the optimization algorithm has not converged for structures with large number of layers. So what do these preliminary results mean? In a way that the results we see is transition, it can be a transition from here, from 20% all the way to 80%, given the fact that we have a difference in absorption of 0.65, which in a sense can be like a 60% difference in absorption. So it could be a difference between going from 20% all the way to 80% in absorption. For the ratio fitness function, so this chart demonstrates the increase in ratio between the absorption of crystalline and amorphous structures resulted by increasing the number of layers. This is the most optimal result produced by the ratio fitness function. The maximum ratio obtained was 6.64, which meant that the absorption for crystalline structure was 6 times bigger than the absorption for amorphous structure. Furthermore, this combination had the maximum difference of 0.66, which is the highest absorption difference we have ever obtained. So to conclude, given the fact that these are preliminary results, we still need to run more simulations as a way to obtain the highest difference in absorption between the two structures, as a way to obtain the best combination of materials and thicknesses. And thank you, Dr. Barones, and this is all thanks to the National Science Foundation.
Any questions or comments? So when I have a question. I mean, you focus on a large range of wavelengths. Is all the visible range the one you want to? I mean, in all your figures, all your plots, you have wavelength in the horizontal. So your optimization is for a range of wavelength, for
No, it's a range of wavelengths. Like, yeah, it's between 400 to 1500.
So next is Carter Williams.
Okay, hi. Thank you for coming to my presentation. My presentation is called Remote Sensing Sediment, Analyzing How Sediment Changes Seasonally in Coastlands of Southeastern United States. My name is Carter Williams, and my mentor is geobiologist Dr. Mariotti. This summer has been an exciting opportunity for me to explore a research field that I was previously entirely unfamiliar with, which is sedimentology. So let's talk a little bit about the importance of the Louisiana wetlands. So the Louisiana wetlands provide many important ecological services, including but not limited to fisheries production, resting areas for migratory species, carbon storage, and water filtration. So though humans rarely do it on purpose, sometimes our actions can inadvertently harm the wildlife and forestry of these areas. So actions made in order to protect the human residents of Louisiana have sometimes resulted in endangering species of animals, lessening water filtration, increased erosion, and other detriments to the wetlands. So in order for us to make sure that we are not endangering any part of the wildlife, we need to take investigative action to understand the environment around us. So what tool can we use to monitor the wetlands? Well, you can probably guess based off the title, but remote sensing. So that would be super useful for monitoring the environment at a macroscopic scale. So what exactly is remote sensing? For those of you who don't know, remote sensing is the scanning of the Earth by satellite or high-flying aircraft in order to obtain information about it. So naturally, satellites are very expensive and difficult to launch, but luckily there is a free resource for scientists out there to access thousands of satellite images. And that resource is called Google Earth Engine. Google Earth Engine combines a multi-petabyte catalog of satellite imagery and geospatial data sets with planetary scale analysis capabilities and makes it available for scientists, researchers, and developers to detect changes, map trends, and quantify differences on the Earth's surface. That is a quote that is taken from their site. This tool would be a perfect fit for geobiologists to analyze the Louisiana wetlands. So let's just take a quick look at the overview of the process of which I'm doing. So first I run a program that is a combination of both Dr. Mariotti's code and my code that is on Google Earth Engine. Then this code takes data from MODIS, which is the Moderate Resolution Imaging Spectroradiometer, which is aboard the Terra satellite, also known as the EOS AM1, and the Aqua satellite, or the EOS PM1, which is really, really cool. And then filters that data based off of time taken, the quality of the pixel, and whether that pixel is relevant. So to see if that pixel is actually relevant, if that is actually on water instead of land, because we're only interested in sediment in the water in this case, I actually take landmass data from another satellite called the Landsat-8, which is also really cool. So next, that data is sorted into a high tide data set and a low tide data set. And finally, that data is exported into a Google Drive where I can then access it in Google Collaboratory to visualize. So Google Collaboratory in this case is kind of serving as the meeting point for all the data that I collect, which is the satellite data, the tidal data, and the streamflow data. So the data that I'm collecting isn't actually in Louisiana in this case. Naturally, Louisiana isn't the only state that would benefit from using satellite imagery and remote sensing. So a lot of tools and techniques that are used to better our understanding of the wetlands can also be used to benefit other coastal states. My mentor, Dr. Mariotti, has specifically tasked me with gathering data of the Altamaha River in southeastern Georgia. So it can be very similar to rivers in Louisiana in some ways. So collecting data about it could prove to be useful in the future. Using Google Earth Engine, let's take a look at the mouth of the Altamaha River. We can organize the pictures based off of tidal prediction data taken from the National Oceanic and Atmospheric Administration, or NOAA for short, so that the pictures are compiled into two separate categories of high tide and low tide. So if we take these two resources and combine them, we'll see what happens. And we get a chart that looks, or a figure that looks a little like this. So here are the average sediment levels for each month of 2019 at both high tide and low tide. So it's kind of hard to see from this distance, but you can make out that low tide has a considerable amount of sediment more than high tide. So let's zoom in just a little bit. Right, and so you can see there's a considerable difference in low tide. There's a lot higher sediment levels. And this makes sense. If you were to pour water into a glass halfway filled with sand, the sand would be disturbed by the motion of the water and be more distributed through the glass. But if the water were drained, the sand would naturally pile up back at the bottom of the glass. So if we were to take this analysis even further and compare the sediment rates during high and low tide with stream flow data taken from the United States Geological Survey at Doctortown, Georgia, we could perhaps see some interesting things. It would be interesting in particular to see how much the stream flow taken a couple miles north of the river mouth affects the levels of sediment. So you can see right here, you can see that little red marker is Doctortown and the mouth is right here. All right. So here's the chart of the average sediment levels per month since 2003. The average for high tide is the blue line, the average for low tide is the orange line, and the average for the river stream flow is the dotted black line. So as we can see, there are a lot of correlations between the river discharge and the sediment levels. You can see one right at 2016, there's one, 2020, there's a big one right around 2010, another one around 2009. So a lot of correlations that can be seen. And here we have another comparison that's further upstream near Broadfield, Georgia. There's only one line plotted for the average monthly sediment because tide is understandably negligible this far upriver, doesn't super matter in this case. But again, we can see many correlations between the stream flow and the average levels of sediment. So thank you so much. That was my presentation.
Questions?
Yeah, I got one. That was an interesting talk, Carter. I was curious how you correlated, I guess, the turbidity or the color of the pixel with suspended sediment concentration in the water. How did you establish that relationship?
So I think the satellite kind of mostly does it for us, but if you're specifically asking about just the way that the color is working, that is just something that I took from Matplotlib. It's just the, and it's also what Dr. Mariotti uses. So I just kind of take that advice from him.
Okay.
Yeah.
Another quick question. So the sediment level and your satellite images, are they open source? Do you have any source we can look it up? Data?
I'm so sorry.
Yeah, so data collection. Yeah, so for data collection in your chart and visualization, and those satellite images open source? Is there any source that we can look it up?
Yeah, so right, Google Earth Engine is open source. I had to apply for it, so it would take a day, but you know, I'm actually not sure. I'm sure that there's got to be a place where it's already visualized out there, but in this case, right, I had to write some code to actually get those images. But Google Earth Engine like has that free for everybody. You just have to apply, which is super fun.
I mean, to the best of my knowledge, all data from NOAA is, I mean, you can access it.
Right.
I suppose from the geological, whatever, I forgot, I suppose that also public, but I'm not that familiar how to access the geological, what is called, reserve or?
Sorry, my internet's a little strange right now, so I can't quite hear as well, but was there a question in there?
No.
Oh, okay.
The website's usgs.gov, and it's all open source.
Okay, and you can access it too. I mean, the data. Okay, no, I'm not that familiar with that, but good to know. Okay, great. There are no more questions. Thank you, Carter. So next, it's Nathaniel.
Yeah, my name is Nathaniel Roble, and this summer I worked on training a quantum convolutional neural network to correctly detect the phase of one-dimensional transistor Ising chains. The transistor Ising model is a popular many-body system that we use to identify phase transitions. And we can define this model as a lattice structure. So we can have a lattice structure of an n number of sites, however many we need. And here we have a representation of a two-dimensional lattice with 9 sites. And each site can be in one of 2 states, +1 or -1 in our case. And right below, we have a Hamiltonian equation. We can use this to calculate the total energy in the system, depending on the orientation of the spins in these specific sites. And we use J to represent a coupling constant. And because these sites in the system can interact with their neighbors through coupling, so we can represent that with J in the equation, and you can see we're summing the total interactions of all our neighboring sites. And the second term in our equation introduces an external magnetic field. And this external magnetic field is going to interact with each site individually. So we're summing those individual reactions as well. We also have our phase diagram, which we can use to plot the phase transitions, where we have temperature on the y-axis and an order parameter on the x-axis. So we're working in a computing environment. So we have 0 temperature. And at 0 temperature, you can see this convergence where we have a quantum critical point. And this is going to signify the phase transition from one phase to the other, with left being an ordered phase at a paramagnetic system and right being a disordered phase with a paramagnetic system. And we use this model in quantum computing. And quantum computing allows us to use quantum mechanical properties to perform computation. Some of these properties include superposition and entanglement. So superposition allows us to put a qubit, the classical analog, or the quantum analog of the classical bit. We're able to put it into a superposition of both the 0 and the 1 state. So it can't be defined as one or the other, but both. And then entanglement allows us to take multiple qubits and entangle them, meaning if we measure the state or change the state of one qubit, it could directly affect the state of a different qubit when they're entangled. So we can't describe these qubits independently of each other. And in quantum computing, we're able to construct quantum circuits. So we can have any number of qubits for our input. Here on the left, we have 2 qubits for our input. And then we enact a series of quantum gates in any order we want. We have 2 here. We have a Hadamard gate acted on the first qubit, so Q0, and then a CNOT gate, which is able to entangle these 2 qubits. And when we construct our quantum circuit, we can change the state of the input qubits to get the desired output. And in quantum computing, we can construct quantum algorithms to solve specific problems. And the algorithm that was used in our data sets is the variational quantum eigensolver. So this algorithm uses the variational method to find close approximations to the ground state of the system. When we don't know the system's true wave function, so we can't calculate its true ground state, we can define a trial wave function. It can be arbitrary or it can be an educated guess. But when we define this trial wave function, we make it depend on some set of parameters. And as we change the value of these parameters, we can notice the calculated ground state for this trial system to go up and down. And when we find that minimum value for the ground state, we know what set of parameters we need, and we have a good approximation to the true ground state of the true wave function. So in quantum computing, we can construct our trial wave function by constructing a circuit. Here we have a 4-qubit circuit with a series of gates acted on them. And we made these gates depend on some parameter values. And when you sweep a range of these parameter values, you can find which set of parameters give you that ground state that you're looking for, so you have your good approximation. And each qubit in this circuit is going to represent a site on the transistor Ising model. So here we have a 1-dimensional system with 4 qubits or 4 sites. And through the variational principle, we know that the approximated ground state that we calculate with our trial function must be greater than or equal to the true ground state of the true system. And using this algorithm, we were able to import a data set. So we didn't do this ourselves on this data set. We just imported it for the purpose of training our neural network, but a little background on the data set that was handed to us. We imported it through TensorFlow. So they were given this Hamiltonian, and G is going to be the order parameter. And this order parameter can range from 0.2 to 1.8 in this scenario. So what they did without knowing the true wave function, they constructed their trial circuit with 4 qubits as well, made their gates depend on some value of the parameter, and then they used a value for G. So we'll start with the first data point using the value of 0.2 for G in that Hamiltonian. So for that value of G, they wanted to calculate, or they wanted to find the set of parameters that give that minimum value for the energy. And then they went to the next value of G. And then using 81 different values of G in this range of 0.2 to 1.8, we were given 81 data points, each representing a trial wave function. And all of these wave functions or circuits are constructed exactly the same as you see here. The only difference is the value for the parameters because of the varying value of G. And G represents or gives us the phase of the system or the label. So any value of G that was less than 1, we have a ferromagnetic system. Any value of G greater than 1, we have a paramagnetic system. And our G equals 1 gives us our quantum critical point where we see a phase transition happening. And another thing on this data set is we noticed that the variational parameters were continuous. So we were able to interpolate the data to create as many data points as we needed, which was very useful in training our neural network. And the quantum convolutional neural network is useful for detecting and classifying quantum data. So what we wanted to do with this neural network was to try to get it or train it to correctly label each wave function or circuit with their corresponding phase. And what we needed to do was we needed to construct a neural network that had a series of convolutional and pooling layers. So the convolutional layers represented at the bottom, its main purpose is to find key information or hidden data within pairs of qubits and send it along by enacting X, Y, and Z rotations on that qubit. The pooling layer tries to take information from pairs of qubits and pull it down to just one qubit and reduce the degrees of freedom. So we can systematically shrink the size of our system down from 8 to 4 to 2 to 1 qubits every time we enact a pooling layer. And once we get down to our final qubit, we can make a measurement on that qubit and find out if the predicted value for the label was correct or incorrect. And regardless if it's wrong or right, we have a value for the loss through the mean squared error, which kind of tells us how far off the prediction was from the true value. And during the training phase, the QCNN uses back propagation to change a set of parameters that are enacted on these specific layers that are on the gates in the layers. And then once it finds a good set of parameters that can make the loss converge, we know that our QCNN is fully trained. And the QCNN is also constructed in a circuit environment. So what we can do is we can just put the wave function circuits on the front end of the series of convolutional and pooling layers. So at the end, we just have one really long circuit that does everything we need. As we input our trial wave function, we go through the layers to take out the important data and pull it down, and then we get our measurement. And I did these tests with system sizes of 4, 8, and 12 qubits or sites, all one-dimensional. And you can see here we have pretty good results for the accuracy in that 99% range. So even as we increase the number of qubits for our input, we still were able to conclude that the QCNN was capable of predicting the phase of the wave functions. And then for our loss, same system sizes, we can see here that the loss is converging as we train the network. And we can see that as we increase the size of the system that we input in our wave function, our loss goes up. And this is expected because we have more input variables, so we're going to have more room for that loss to grow. But at the end of the day, you saw that our accuracy was still high. We still got that 99% range for our system sizes of 12. So regardless of this increase in loss, we know that our system or our network was capable of predicting the phase of systems for each size. This is a summary of the results that I just discussed. And from this, we concluded that the QCNN model is a feasible method to classify quantum data using this model. And from here, we would like to push the limits of a quantum convolutional neural network by implementing more complex systems. And some ideas would be to use the same model with more complex Hamiltonian equations. We could test our neural network using different many-body systems, or we can construct our own neural network model for testing and generate our own data sets using the variational quantum eigensolver. Questions?
Well, Natalie, I would like to know, I mean, you didn't know about all this at the beginning of the program. So what has been your thought going through these last 10 weeks?
My thoughts through the program is I definitely enjoyed it. I didn't know anything about quantum mechanics, coding, machine learning. So I definitely learned a lot, and it was difficult at first. But I can say overall, I really enjoyed the program and the research that I was involved with, as well as the mentor, Dr. Coming or Professor Coming, that helped me through this research. So I'd like to say thank you to him as well.
Jonathan, so whenever you are ready.
Good morning, everyone. My name is Jonathan Wu, and I am an undergraduate student at the University of Pennsylvania studying engineering and economics. This REU program, I worked with Dr. Michael Brilinski, who is the head of the Computational Biology and Systems group at LSU, and he's also a part of Center of Computation and Technology. My project title is A Deep Learning Approach to Breast Cancer Detection Based on Sequences of Single Nucleotide Polymorphism Data. So to really understand my project, we have to understand what a single nucleotide polymorphism, or as I'll be referring to this presentation as SNP. They're the most common type of genetic variation among people. Each SNP represents a difference or variation in a single nucleotide base pair. So we all know what DNA looks like. It's a double helix of many nucleotide base pairs in a wound up strand, like we can see in the top right corner. A SNP is when in one of these locations of base pairs, there's high variation among what's actually present. So we know there's 4 basic base pairs: guanine, cytosine, adenine, and thymine. And a SNP is in when a single location, like if we can see here, it's not really a uniform assessment of what's going to be there. So we can see on the left of this, it's all adenine, and then the right is all guanine, but in this particular location, it's distributed randomly. So we can use this to our advantage to classify breast cancer. First, I would like to talk about the data set that I'm using, which is from the National Institute of Health. We have 28,281 patients, and the data we have is the DNA sequences for all 23 chromosomes for each patient, because humans have 23 homologous chromosomes. And we also have the status of whether these subjects have breast cancer or not. We can see in this bar chart on the right that the data distribution is relatively equal, with about 14,000 not having breast cancer and 14,000 having some sort of breast cancer, whether that's an in situ case, a case with unknown invasiveness, or an invasive case. Just for simplicity, we classified all 3 of these cases as having breast cancer. So how did we format the data to actually input it into our deep learning model? Well, this is an example of how I represented a SNP, a single nucleotide polymorphism, as a representation of part of a sequence representation. So the first number here represents the distance between this current SNP and the previous SNP. This letter here represents the major allele, or the base pair that would usually be found in the population, or the majority base pair. And then this is a minor allele, or the base pair observed in the patient. So what the SNP changed the base pair to in this particular location. And in sequence format, this is what it would look like. So we have our base pair distances separating the 2 SNPs and your major and minor alleles. And this will be across 1 chromosome. So 1 chromosome can have as many as 20,000 SNPs and as little as 2,000 SNPs. So there's high variation in the amount of SNPs present in each chromosome. And one problem that we encountered here was we can see this set has very high cardinality, meaning that we can see these numbers are very unique. For example, the number 29,753 most likely wouldn't appear more than a couple hundred times out of hundreds of millions of SNPs that we encountered, and the number 16,333 would have the same. So what we had to do is use a quantization method to make sure that our base pair distances had a higher number of frequency or occurrences. And the way that I visualize this, or the way that I went about quantization, is with a 2D histogram. So on the x-axis, we have the number of nucleotide base pairs between SNPs, so the base pair distance, as noted before. And on the y-axis, we have the number of occurrences, so the number of times these numbers occurred. And we can see it that at a high number of base pair distances, so when a SNP is separated by millions or hundreds of thousands of base pairs, the number of times that they repeat itself isn't very high. Whereas in a lower number of base pair distances. So when SNPs are separated by maybe 10, 20 base pairs, this happens quite often. So what I did is I didn't touch anything from 1 to 1,000. I kept those distances the same, but for anything above 1,000 all the way to 1 million, I quantized them into buckets. And so we can look at the statistics or the results of how this quantization went. The vocabulary size decreased drastically as I quantized each higher base pair distance into buckets. So there's only 2,000 distances rather than 1,190,000. The average number of occurrences per these base pair distances went up by a lot, as before it was only 3,000 and afterwards about 1.77 million. And the 25th percentile of occurrences as well as 75th percentile of occurrences went up by a large margin as well. And after I quantized these sequences and put them into a good format that I believed would work, what happened next was called tokenization. So of course, machine learning models can't just read these DNA SNP sequences as is. There has to be a process to feed into the transformer or machine learning model so that it can process this data. So what happens is that you input a string of the whole sequence of all 2,000 to 20,000 SNPs, however as much is in that chromosome. And afterwards, the tokenizer will break this whole string up into words. So each of these would be considered a word or a token into the model. After it splits it up into a list of words rather than a single string, it'll convert these words into input IDs. So it'll start from the start, and each unique value will be assigned a new input ID. Notice how it said unique value. So if a value of a word or token repeated itself, like in this case, 29,000 AG, 29,000 AG, it would just resort to the previous input ID that it was assigned before. So basically, this is a mapping to a unique vocabulary so that the machine learning model can read it feasibly. So how did we actually go about training and classifying for breast cancer? We used what's called a state-of-the-art machine learning model called the transformer, and I use a specific variant called the longformer. Basically, transformers are what Google uses for Google Translate or next sentence prediction or sequence to sequence tasks. So when you want to translate a sentence from English to Korean or English to Japanese or Chinese, what will happen is it's called an attention mechanism that you iterate through every single word. And these words attend to each other. So they gather input from each other to find out what the correct word would be as the translation. And this happens for next sentence prediction as well. For example, if you're in a Google search and you're looking something up, it's going to use attention to try to figure out what you'll type next. So the longformer is a variant of the transformer because, as I said before, we have upwards of 20,000 words in a whole sequence, right? 20,000 SNPs per sequence. So the attention mechanism, because it attends to every single word, it's very computationally costly, with the memory complexity being n squared, with n being the number of tokens in the sequence. So longformer uses an attention window, which a word won't attend to all of the other words in the sequence, but it will attend to a certain number of words before and after, which will allow for less memory complexity. And so the pre-training results of this worked quite well, with the loss starting at about 9.5 and going all the way down to 4.8 with about a 50% reduction. And the reason why I'm saying pre-training is because, as I said before, transformers only handle sequence-to-sequence tasks. So if you're translating a sentence or if you're predicting what happens next in a sentence or if you're answering a question. So you can't train it directly for breast cancer classification at the start. The task that I use to train for pre-training was called masked language modeling. So what happens in here is that 15% of the input words or input tokens are masked, and the language model will try to predict what these masked tokens are. So you basically hide 15% of the words randomly, and then the model will try to predict what these 15% of the words are. And after we achieved this, we went on to fine tuning for the actual output classification. So what happens is that these input and these hidden layers in the deep neural network had already been trained from pre-training. So we can freeze these, meaning that they're not going to be trained during fine tuning. The only thing that's being trained is these output layer, which determines whether someone has breast cancer or not, with two output neurons, one for breast cancer and one for not having breast cancer. And we used five-fold cross-validation to gather the accuracy of our model and found that it's actually 64%. Now, normally in binary classification, for as far as the deep learning model goes, this isn't very high. But I would like to point out the fact that we only actually used data from one chromosome for this project due to the limited amount of time. In the future, we want to create a model that uses all 23 chromosomes to predict whether someone has breast cancer or not. And we hope that the more information we have will lead to better results. So I would like to thank Dr. Moreno and my mentor, Dr. Brilinski, again for supplying me with all the resources and knowledges for this REU program, and I'll take any questions now.
That was really interesting research. I was curious about your last comment, Jonathan. You said since this is 64% accuracy and eventually you want to develop more, but how long does it take? Why you didn't try this time, and how long does it take just a physical time if you want to go through all?
Yeah, so that's actually something we're working on right now. So like I said before, we have 28,281 patients, and for one chromosome, right? So you have, that's basically you have 28,000 inputs data. If you want to do all 23 chromosomes for pre-training, then you're feeding in 28,281 times 23. So you're basically increasing your training time times 23. And for pre-training, I'll just say it took about 48 hours. So 48 hours times 23, it's gonna be over 2 months, right? So what we're trying to do now is, instead of using all 28,000 patients, we're going to only use about 2,000 of them. And instead of using 23 chromosomes, we're gonna only use 10 of them. So theoretically, 2,800 patients should still be enough. And because we're decreasing the amount of patients by 10, right? But then we're increasing the amount of chromosomes per patient by 10. So it should still only take 48 hours. And this is something I'm actually going to try this week. And hopefully we're not sacrificing, or we're actually going to improve in performance, even though we're sacrificing a good bit of test subjects in that.
Thank you.
Yeah, no problem.
I have a couple of questions. I mean, what advantage gives you to use the transformer? I mean, can you just use the whole neural network to classify the?
Yeah, so actually, before I came in, they already tried a vector representation. They tried convolutional neural networks. They also tried what's called genomic signal processing, and all of these methods failed. So this is kind of the 5th or 6th iteration of us going through this data set and trying to find a method that'll actually work. So the reason why transformer works, in our opinion, of course, there's still a lot we don't understand because this hasn't ever been done before at this large scale to actually predict whether someone has breast cancer or not. Sometimes they'll say the risk of having breast cancer, but they can't actually definitively tell. We use transformer because of it's just the newest technology in terms of deep learning, and it can handle the most amount of input data in this sequence format. So DNA was perfect for that.
And why you use 2 output instead of just 1? Because to classify, I mean, you could use just 1. Why you use 2?
Right. So you're also right about that. The output could just be one neuron, and that neuron could whether tell if it's like sigmoid, right? So if it's less than 0.5, that means you don't have breast cancer. If it's greater than 0.5, that means you do. And I actually did try this approach. So I did use a sigmoid output, and then that would be represented as probabilities as either one or binary, right? But this didn't work as well as the cross entropy loss that we use. And I'm not completely sure why. I did try both of these approaches, but with 2 output neurons and then using cross entropy loss instead of binary cross entropy loss, and sigmoid worked better for some reason.
Okay, yeah. I'm curious to understand the reason.
Yeah.
Just quick, just out of my curiosity, have you thought of image processing?
Yeah, so that was an approach we went with see a convolutional neural network would be image processing. And that's something we did try, and one of my lab members tried before me and they said it didn't work. So I haven't personally tried it myself, because at the start of this, we weren't too hopeful either about this representation. It took me almost the entirety of REU to even get it to have some sort of performance, like better than 50-50. So it's just a problem. This, like the SNP problem, is just something that's never really been done before. So maybe convolutional neural networks could be a solution if we spent more time, but that's just something I haven't personally done yet. And that's something that my lab partner said didn't work, so.
Okay, so I will really like to thank everybody. I mean, you all did a wonderful work here, well, here at your places, but virtually here at LSU. Yeah, thank you very much for everything you did.